Hello and good evening, everyone. My name is Cynthia Mutisya. I'm from Group 44, and we are presenting on engineered probiotic bacteria to prevent malaria infection in humans. And these are my colleagues. We have Pamela, Innocent, Sherephine, and I. Next slide. Next slide, please.
And to move again. So
I'll be doing a bit of background information on malaria. And as we all know, malaria is a vector-borne parasitic infectious disease that has a significant role in morbidity and mortality in many nations. So malaria is caused by a parasite called the Plasmodium, and we have 5 of these parasites. We have the Plasmodium knowlesi, Plasmodium ovale, Plasmodium malariae, Plasmodium falciparum, and Plasmodium vivax. We have Plasmodium falciparum being the deadliest malaria parasite, followed by Plasmodium vivax. So according to WHO information that we've gathered, in the year 2022, an estimated number of malaria deaths stood at 608,000 people, with most of these people being affected from the sub-Saharan population and the tropical regions. So malaria can be prevented by use of vector controls, chemoprophylaxis, preventive chemotherapies, and use of vaccines. And also an early diagnosis and treatment of malaria prevents death and reduces the transmission rate of malaria. Next slide. On the life cycle of malaria, we have 3 stages. We have the gametocyte stage, the sporozoite stage, and then merozoite stage. In the gametocyte stage, both the male and the female eggs mate inside the gut of the malaria to produce the sporozoite, which moves to the salivary glands of the malaria, of the Plasmodium, And when the mosquito containing this Plasmodium bites an individual, it infects them with the sporozoite, and the sporozoite will move to the liver, which is going to grow into a merozoite. The merozoite is then going to attack the red blood cells in the individual, and this will cause an individual to have the cold chills, a common symptom that we normally see with individuals who have malaria in their lives. So once an infected mosquito bites on this individual who's having this Plasmodium in their blood, it is going to ingest on the eggs, and these eggs are going to multiply inside the gut of the mosquito, and the life cycle starts again. Next slide. So during this migratory phase, infective sporozoites evade the host immune system, transfers multiple cells to cross the hepatic sinusoidal barrier, and invade a suitable hepatocyte via interactions with select host receptors like the cell surface heparan sulfate proteoglycan and the glypian-3, the GPC3. The extracellular sporozoites are said to shed cell membrane vesicles covered with CSP and the trap, which trigger the host immune system. Malaria liver stage, with a bottleneck in parasite load and reduced clinical symptoms, represents a vulnerable therapeutic target to prevent progression to clinical disease. However, the few drugs that target the liver stage are lethal to patients with a glucose-6-phosphate dehydrogenase deficiency, emphasizing a need for improved development of safe drugs treating malaria. I'll be inviting Innocent to proceed from there.
Thanks very much. I'm here by the name of Innocent. I'm going to talk about why you have chosen lactobacillus and its competing technology. Advantage of lactobacillus. Lactobacillus is preferred because they are involved in several functions such as defense against pathogen, maintaining mucosal immune hemostasis and intestinal integrity. Is resistant to gastric juice, pyruvate, and pancreatic enzyme in the upper gastrointestinal tract. Also, they have proven ability to reduce endotoxemia and severity of experimental alcoholic liver diseases. Lactobacillus plantarum MH2 decrease both liver cholesterol and triglycerides, and also restoring neutrophil function in alcoholic cirrhotic patients. They are safe to treat patients with defective immunity, also lack of adverse effect on markers of inflammation infection. They also have a genome that can be designed and being completed by inserting new genes. Competing technology. Symbiote-based transmission blocking strategy, what we call paratransgenesis, occur by engineered natural plasmodium blocking symbiotic bacterium used include Pantoya species and Serratia species, which drives the effector protein lipase into the mosquito gut to Plasmodium parasite via host serum-induced extracellular vesicles, leading to targeted killing of the parasite. Thanks very much. I'd like to invite Sherephine to continue with the next slide.
Okay, my name is Sherephine, and I'll be taking you through the advantages of our main of other compounds. So the first one is biological activity. Okay. Hello again, my name is Sherephine Achieng, and I'll be taking through the rest of the session. I'll be taking you why we choose a main compound over other compounds. So the advantage number one is because of its biological activity. Amine is good at fighting malaria because it can stop malaria parasites from growing. Number two, selective toxicity. Amine is special because it can kill malaria without harming cells. The third one is ability to cross biological barriers. Amine is able to cross blood-brain barrier, which means it can reach and target parasites in different parts of the body. The fourth point, because of its natural occurrence, since amine is found in some food, suggests that it's safe for human and to be easy to develop drugs. Next slide. So here in this slide, I'll be talking about the production of amine. So we start with tryptophan, which is the building block for many important compounds, which will be turned into tryptophan with an enzyme called tryptophan decarbonylase, which forms to tetrahydrobetacarbolin, which later forms a more complex structure, forming the amine. The evidence from research can be found from plant-based synthesis, which the link is over here, and the second one is from microbial production, and the link is also here. Thank you.
Hello guys, my name is Pamela. So in our methodology, Sherephine stated, so we can see the method, it will involve the DPT cycle, design, build, and test cycle. And in our pathway engineering cycle, it will involve identification of the functional enzyme, HAMEN, via retrosynthesis pathway using RetroPath version 2.0 workflow, or a computational approach utilizing automated plant smash. HAMEN will also this HAMEN will be isolated from mushroom, Psilocybe cubensis. This is one of the sources, plant sources of this compound, to be isolated via protein fractionation, and it will be identified using protein mass spectrometry followed by transcriptomic or genomic mapping. The biosynthetic pathway genes that have been reported in published articles in the L-tryptophan pathway include L-tryptophan decarboxylase, Escherichia coli tryptophan synthase. Then also we have a kinase. So these were reported in a psilocybin, a compound from this mushroom. These biosynthetic pathway genes will all be codon optimized for Lactobacillus plantarum and chemically synthesized by GenScript. Our vector construct, which is our probiotic Lactobacillus plantarum, will include the vector, Lactobacillus plantarum vector. This is a broad host range shuttle vector, which is used by most Lactobacillus species. It will also consist of 2 plasmids, each containing an origin of replication, antimicrobial, antibacterial resistant gene, and a reporter. In our case, in this method, we expect to use gas A reporter and superfolder green fluorescent protein reporter. The construct will also consist of TAF promoters, elongation factors, ETAG. This is a HAS-A gene isolated from E. coli hemolysis. It's a heme binding protein secretion system. So all the plasmids will be constructed by Gibson or Golden Gate assembly. The HAMEN biosynthetic pathway genes will be cloned onto the multiple cloning sites of the lab vector. Plasmids harboring pathway genes will be used as PCR templates to obtain donor DNA fragments with a 40 base pair homology arms to the integration sites in lab genome. Our strain construct, which is our probiotic lab bacteria, Lactobacillus plantarum, and now it will be modified, and all the modifications of this lab genome will be based on the CRISPR-Cas9 double-stranded DNA recombination. The single guide RNA spacer sequences will be designed by Benchlin and then cloned into the BSI sites of the lab vector. The single guide RNA plasmids and donor DNA fragments will be co-transformed into the recombinant lab competent cells. The integration of the heterologous gene expression cassettes will be verified by colony PCR and further confirmed by DNA sequencing. The strategies that will be used, or some of the strategies that will be used for the engineered lactobacillus to produce these HAMEN compounds will include a natural substrate feeding, combinatorial biosynthesis, as well as novel enzyme incorporation. Thank you from Group 44.